Now we discuss about sources of drugs. First, see the pharmacology. It is a science of drugs. It covers all aspects of the knowledge about the drugs. First, what is the definition for the drug? According to the WHO organization, any substance or product that is used or intended to be modify or explore physiological systems or pathological states for the benefit of the recipient. Next is the drug nomenclature. Usually the drug has three names: chemical name, non-priority name and priority name. Non-proprietary name is a generic name, proprietary name is a trade name or brand name. Any manufacturer can make own brand name. Non-proprietary name, proprietary name. First we will see the chemical name. These are the given according to the chemical constitution of the drug and chemical structure base మీద we will give the chemical name. Chemical gives exact chemical composition of the drug, places atoms and molecular structure. Here we will see the different colors, red and blue, how it is arranged, means that bonding. Next is a non-proprietary name, is the official name. It is assigned by the United States of Adopted Name Council. It is uniform for all over the world. Is typically derived from the chemical name, usually shorter one. Example means aspirin, paracetamol. Next is a proprietary name. It is given by the pharmaceutical manufacturer. So, trade name, otherwise we call as a trade name. Name is registered by the manufacturer. Trademark symbol only be used by the single manufacturer. Same drug may have several trade names. The first letter of the name should be capitalized. Here see the one example. Chemical name is acetyl salicylic acid. Non-proprietary name is aspirin. Trade name is different, Disprin and Bayer's aspirin in USA and Ecospirin. Now it is marketed as Ecospirin 150, Ecospirin 75, used as the antiplatelet drug, and before it is used as an analgesic drug. Now it is banned, it is used now only antiplatelet drug. Next is the source of drugs. Usually the drugs are sources from natural or synthetic, natural plants, animal, microorganisms and minerals, and synthetic means is semi-synthetic and synthetic. Semi-synthetic means half synthetic and half natural. And plant sources. Plant sources means leaf. Example, digitalis drug is digoxin, used for the congestive heart failure. And bark, cinchona, drugs quinine, used for malaria. Fruit, opium, drug is morphine, used for analgesic. And seed is azarin, and drug is anticholinesterase and used for MG means myasthenia gravis. And some more examples, cinchona bark is a trade name is quinidine and under the classification of antiarrhythmic and malaria. And purple foxglove, that means digitalis, is used for cardiotonic. Poppy plant is a paregoric, morphine coating, antidiarrheal, analgesic and antitussive. You can use it. Nicotiana tabacum is a nicotinamide receptor stimulant drug. Atropa belladonna is an anticholinergic drug used in the opium poison. Opium seed is a morphine analgesic. The natural drugs have different categories to be used. Here we see the morphine preparation. These natural sources opium poppy, and crude drug preparation. Opium is a dried juice of the poppy seed capsule, and pure drug compound is morphine is extract from the pure drug, and pharmaceutical morphine is sulfates, tablet, oral solution and solutions for the injection. We can use it. And from natural source we extract the crude drug and then purify it, and then from purified we can prepare different categories, tablets or injection. Here is vinca rosea. It contains vinca alkaloids, used for anti-cancer. Digitalis purpura is used for CHF. Cinchona pubescens used for malaria. Revolvia serpentina drug is risperidone, used for the antihypertensive agents. These are the drugs are all people are studied in pharmacognosy. Important pharmacological active principle in plants are alkaloids, glycosides, oils, resins, gums and tannins. These are the pharmacological active drug molecules. This active moiety we have the different action. First we will see the alkaloids. The alkaloids are nitrogenous heterocyclic bases insoluble in water and form salts with acid which are soluble in water. Here drug is atropine and quinine. Plant atropine is from Atropa belladonna, quinine is coming from the cinchona bark. Both are alkaloids. And some more alkaloids we can see the and morphine, papaver somniferum, reserpine, revolfia, serpentina, nicotine is coming from tobacco leaves. Alkaloids names usually end with the ine. See morphine, reserpine, nicotine, atropine is ends with ine. Next is glycosides. These are the combination of sugar with other organic structures. Glucose and non-sugar moiety will get the glycosides. Sugar portion governs the pharmacokinetic characteristics of the glycoside. On hydrolysis with mineral acids, all glycosides split up into sugar and non-sugar residues. Cardioglycosides, digitoxin, digoxin is obtained from the foxglove leaves, Distadis purpurea. So, in this sugar molecule we have the active moiety, it gives the pharmacokinetic parameters of the drug. Next is oils. Generally 3 types of oils are used for medical purpose: essential oils, fixed oils and mineral oils. Essential oils means volatile oils. If it is example is eucalyptus is a volatile oil. Fixed oil is groundnut or castor oil. Mineral oil is groundnut oil. See, essential oil is the volatile oil obtained from the leaves and flower petals by steam distillation. Terpene derivatives, so these have a aroma, steam, volatile, no food value, do not form the soaps with alkaloids, they do not rancid on prolonged stay, and they won't be destroyed. In long period ఉన్నందుకు దాని యొక్క aroma అనేది పెరుగుతూనే ఉంటుంది. And uses, it can be used as a carbonative, antiseptic and flavoring agents and pain relieving agents. For carbonative means for expulsion of gas from the stomach. Example, ginger and eucalyptus oil. Antiseptic is a mouthwashes. Flavoring agents, peppermint oil. Pain relieving agents, clove oil for toothache, act as a counter irritant. Next, fixed oil is obtained from solvent extraction of the crushed seeds. Usually triglycerides, saturated from the animal, unsaturated from the plant. These are the non-volatile, have a caloric value and calories వస్తాయి వీటి నుంచి. We can form the soaps with alkaloids, become rancid and prolonged stay. And long stay ఉంచితే కనుక ఇవి rancid అంటే డిస్ట్రాయ్ అయిపోతాయి, వాటి యొక్క నేచర్ అనేది కోల్పోతుంది. ఎగ్జాంపుల్ గ్రౌండ్నట్ ఆయిల్, కోకోనట్ ఆయిల్, ఆలివ్ ఆయిల్ and castor oil we can use it as a purgative. Next is the mineral oil, we can obtain from the dry distillation of the wood. Example is liquid paraffin, hydrocarbon derived from the petroleum. Usually liquid paraffin you can use it as a lubricant laxative for promote defecation. Liquid paraffin you can use it in cardiac patients. Next is the resins. These are the polymers of the volatile oils and insoluble in water. Example is benzoin, used as a inhalation in common cold and you can treatment in cough. బెంజోయిన్ మన ధూపం వేస్తూ ఉంటారు కదా, ధూపం, దట్ ఈస్ రెజిన్. యూజువల్లీ యూ కెన్ యూజ్ ఇట్ ఫర్ ది కోల్డ్. Next is the gums, secretory products of the plants. These are the dispersible in water and form adhesive mucilaginous colloids. Example is gum acacia, used as an emulsifying and suspending agent. In syrups making lo we emulsifying agent ga లేదా సస్పెండింగ్ ఏజెంట్ గా కూడా యూస్ చేస్తారు. సిరప్స్ కానీ లోషన్స్ కానీ ప్రిపేర్ చేస్తాం కదా. నెక్స్ట్ ఈజ్ ద టానిన్స్ ఈజ్ ద నాన్ నైట్రోజినస్ ఫినాలిక్ డెరివేటివ్స్ ఫ్రమ్ ప్లాంట్ ఈ సాల్బుల్ ఇన్ వాటర్ usually used as an astringent. It's precipitate the surface proteins. Astringent action చేస్తే once we apply on the surface of the skin it shows the tingling sensation. Example turpentine oil కూడా యూజ్ చేస్తారు tannins లో. And animal source వచ్చేసి obtain the drug from animals is the heparin, insulin, thyroxine, vitamin B12, cod-liver oil, antitoxic serum. Usually animals ఇక్కడ leech, port, pancreas, thyroid, liver extract, మన హ్యూమన్ థైరాయిడ్ అండ్ లివర్ ఎక్స్ట్రాక్ట్ నుంచి కూడా మనం ప్రిపేర్ చేయొచ్చు డ్రగ్స్ని. Here some of the examples: pancreas of the cow, insulin, regular classification under anti-diabetic hormone, and pancreas pork, and stomach of the cow, pepsin and extract చేస్తారు, digestive hormone, thyroid gland of the animal, thyroid, it used as a hormone. And microorganism source, bacterial, fungi, molds are the important source of many life-saving drugs. These are obtained from the microorganisms used to kill the microorganism. Drugs వచ్చేసి penicillin, chloramphenicol, griseofulvin, streptomycin and neomycin. Penicillin, Penicillium notatum, chloramphenicol, streptomycin veniculae, and griseofulvin, Penicillium griseofilium. Streptomycin is Streptomycin griseus, neomycin is Streptomycin fradei. And then mineral sources used in pharmacotherapy, minerals are ferrous sulfate used for anemia, and magnesium sulfate used for purgative, and sodium bicarbonate is antacid, and aluminum hydroxide is also used as a antacid. These minerals we people are studying in pharmaceutical and organic chemistry. Microorganism sources we studied in the biotechnology. And then other mineral sources, magnesium, trade name is milk of magnesia, used for antacid and laxative. Zinc is zinc oxide ointment, sunscreen lotion and skin protectant ga use chestar. And gold mineral అనేది solgnal and arnofin and trade name తో వస్తుంది, anti-inflammatory, used in treatment for the rheumatoid arthritis యూస్ చేస్తారు. These are all the natural one. Next is a synthetic. Presently majority of the drugs are obtained from synthetically. Some of the drugs which are earlierly obtained from the plant and today synthesized in the lab. Advantages: quality can be controlled, process is easier and cheaper, more potent and safer, and we can prepare the large scale production. Some of the drugs is meperidine, trade name is Demerol, used for analgesic, and diphenoxylate, Lomotil, antidiarrheal, and cotrimoxazole is the trade name is Septra, is used for anti-infective, sulfonamide, used in the treatment of the urinary tract infections. And then some semi-synthetic products also there. These are the mainly obtained from the changing the chemical structure of the naturally obtaining drugs. Here example is atropine bromide and penicillin substrate by changing the R side chain. Already natural one, but we just modifying the branching. If modifying the branching, that means alkyl groups or any substitution, we can make new compounds. Next is a human source. HCG is obtained from pregnant women, monotrophin, postmenopausal women urine, regular insulin human, urokinase, human kidney cells, and recombinant growth hormone is obtained from the human. HCG means human chorionic gonadotropin. And a genetically engineering technique. A relatively new methodology involves the blending of discovery of from the molecular biology or DNA technology, DNA alteration and gene splicing and immunopharmacology. Example is hepatitis B and insulin, human insulin of the recombinant DNA techniques. Some recombinant DNA techniques ద్వారా మనము drugs కూడా ప్రిపేర్ చేయొచ్చు. First, formation of genetically engineered hepatitis B vaccine. Genetic material is first extracted from the hepatitis virus, and gene that direct production of surface of protein is located. This gene removed from the viral DNA and inserted into plasmid. The plasmid are then inserted into yeast cell. Yeast is grown by fermentation. After 48 days, yeast cells are ruptured, then extracted surface protein, purified and stabilized, and hepatitis B vaccine is ready for use. So this is the, thank you for watching. Please subscribe to our channel.